a lot with supply drums. So this can come from anywhere in the world into a facility. And we have to be sure that the drum's clean, the product's double bagged. When we open that drum, there's going to be no contamination risk for the operator. It's really, really important that we understand how that material's coming in. So in this case, the system's actually failed because we've failed at the starting point where the material is contaminated or the drum is contaminated. So now that will transfer through the complete process. So another example, same kind of thing. Again, the right containment system. This time the drum's correct. We've got a clean drum and we've got a managed drum. We've got the right outlet solution, but we then transfer that away from the isolator and we need to get that product into the process. If that's not carried on into the process, then we're going to have a failure further down the line. So when you're charging your powders into a vessel or when you're transferring those powders into the next step, that has to be considered as well, because you're transferring that containment solution all the way through the process. So it's very important to really think about the bigger picture rather than just that piece of equipment in the center of the screen there. So obviously we need everything ticked, and there we go. We've got a perfect system and a perfect solution, and we can maintain the containment throughout. Very simple slide, very simple visual, but please think about this when you're designing your systems and designing your solutions, and you're coming out to people like ourselves for a containment isolator or a downflow booth. Think about the bigger picture. So there we go. We're all passed and we're good to go. So any questions at the moment before I move on? Maybe we'll put those towards the end of the presentation. So now I'm going to go through the product range, and as I said previously, you know, we're a bespoke business. There are applications that you may not see on here, but it doesn't mean we've not done those systems. So we have three ranges within our containment group. So we have the WorkSafe range, and this is predominantly the sort of downflow booth technology, so the open handling. We then move to the UltraSafe range, and this is the containment isolators or glove boxes, and this can vary from different containment levels, but generally for OEB 4, 5s, and 6s. Although there are applications for lower OEB bands, but there might be some sensitizers. The materials might be harmful for other reasons. So these are the kind of things we need to think about as well. And then we have our EnviroSafe range, which is the aseptic part of the product range. So we're going to take you through the examples for each one. So as we move into the WorkSafe, the main product really is a downflow booth. This is potentially the first containment solution that was developed many years ago. And this, I call this the workhorse. This is where quite a lot of the materials are handled within a warehouse scenario, within a dispensing scenario. This would give us typically OEB 1 to 3. But on the right-hand side there, we now can include high containment screens. So as you can see there, the operator will work behind the screen and carry out the various processes. This will then give us another level of containment, so we can potentially move that to an OEB 4 application, but we have to think about what the process involves. We also supply these booths what we call screen ready. So in the future, you can bring in a high containment screen if you didn't then move into more high containment operations. A smaller version of the downflow booth is the downflow bench. So this was developed for laboratories and for R&D. So it's using the same principles of the booth with HEPA filtration in safe change housings, 90% recirc. But in this instance, we're operating through a working slot or a working space. And that allows us to do small scale operations, typically 1 kilo. We wouldn't recommend going into a higher volume because, as you can see, it would be very difficult to handle. We've just tested this system. We've done an independent containment test, and we were getting figures way below 1 microgram. But we have to be cautious because this has not been designed for a high containment system. All our systems, by the way, can be developed for ATEX environments, so your zone 21, zone 22, zone 1, zone 2, and that can be a combination of ATEX and safe area. Again, these are quite important bits of information that we need with the design phase.
Paul,
sorry, yeah.
I think there's a question from Krishna Gupta, sir.
Okay. Can't quite hear.
Yeah, Krishna, sir, do you have any query? Krishna, sir? Krishna Gupta, sir? I think...
maybe we can look at that at the end as a chat or something.
Yeah, right.
Okay, but if he comes back in, just let me know. So just to move on to part of the product range and the work safe. So these are the Kilo Labs. These are becoming more common now, where we're looking to handle process reactors. We're looking to handle solvents within a contained environment. So this system is literally a single pass design, and this allows the operator to work through within these doors that you can see on the screen. There are operating slots. So you can work through a slot, and it gives us 0.5 meters per second protection from outside to in, and that allows interfaces with I don't know, valves and sample systems, etc., within that reactor. So these are becoming more and more common now because the materials that are being handled are obviously becoming more hazardous. And then we move into another technology which is becoming more common. Drum transfer booths. There's a regulation called REACH, which is the evaluation of hazardous chemicals. And that regulation is now asking for drum handling, which was typically done within an open environment within the facility, to be enclosed because of the potential risks of exposure to the hazardous compounds and vapors. And there's also a potential explosion risk, because this is potentially 50 litres of material within those drums. So we're now housing these drums within enclosures, again similar to the Kilo Lab, where we use the single pass design. And this particular design now incorporates what we call a liquid safe transfer system. So that allows us to dock that lance into the drum. It's a cleaner system. It takes the elements of handling a contaminated lance or a contaminated hose within that system, and then we utilize pump technology to transfer from the drum into the next part of the process, which could be most of the time it's a process vessel or reactor system. So again, it's just taking that envelope and designing that around the drum transfer system. When it comes to handling mainly solvents, there's a risk now of material contamination and compatibility. So an option we can provide is to actually coat the material. So this would be a stainless steel construction typically, but we can now coat that material with what's called an armor coat, and this will give us improved resistance from a number of chemicals. We've had this tested with phenyl chloride, for example, which is one of the nastiest compounds that's used within the pharmaceutical industry, and this will give us better protection of the system and avoid any corrosion effects. And it's also a competitive solution versus Hastelloy, which would be the next obvious choice for an improved material construction. So again, we can coat an isolator, we can coat the booth, we can coat any system really. This was just an example of a drum booth that we provided recently. And then the pack-off systems, again, these are quite common in the manufacturing areas where you're coming from a filter dryer or a centrifuge. So we're packing off using the continuous liner system. So we have an inner bag and an outer bag. So again, this gives us a contained solution using the continuous liner methodology to pack off those powders. So then we move into the ultra safe range. So as I mentioned previously, these are the glove box isolators. This is probably our biggest product range. We do more isolator systems and downflow buoys. We focus on the real high containment applications. Before we go into the detailed design and manufacturing, we will always do an ergonomic mock-up. In my opinion, this is probably the most important part of a containment project for isolators, because it's at this stage when the operators will be involved, when we can assess all the ergonomic operations. We can look at handling materials, we can look at handling vessels, drums, anything that's part of the process, we would then replicate within the mock-up. And this is really important, because if you get it wrong at this stage, then the system will fail further on because it's not workable or usable. And again, one of my biggest recommendations is the operators get involved in this process from day one. If they like the system, they'll use it. It's as simple as that. And their input's valuable, because they're the people that will be working on these systems day in, day out. This particular, because of the COVID scenario, these mock-ups are shipped to the customer site. So because of the travel issues, we can now ship these to site. The customer will do the review to our guidelines and then feed them back into the design. An alternative solution we've used recently is using HoloLens technology. So this is a camera system which will sit on the engineer's head, and as you can see by this video, the information is relayed through the camera, and it allows the operator on the other side of the world to actually see the process. So this is our engineer carrying out the task. There's a lot of visuals here, but there's also the opportunity where the customer can speak direct to our engineer and instruct them on the process. So in this case, they're working on connections to a vessel. This is actually within one of the Kilo labs that we talked about previously. So we can try and assess all the various key points using the HoloLens technology, which has been used quite a lot over the last 12 months, as you can expect. So we don't want to hold the process up. We want to get that program moving. And we don't want COVID to get in the way. So this has been a real powerful solution. So different types of systems. So I'm just going to go through a number of different applications. So starting off with laboratory isolators. So this is a combination really of small scale operations. Again, it's R&D. This particular example is showing an interconnection with a fume cupboard. So we can carry out liquid chemistry, and we can transfer that liquid chemistry into the isolator for any solid handling and any compounding. This particular system also utilizes vacuum tray dryers. So at the top right there, you can see the tray dryer has been integrated into the isolator, so we can then transfer a slurry into the chamber, do the drying process, and then that will come out as a dry powder, and we can handle that safely within the isolator. You'll see the word integration on the screen. That's a word I'm going to use quite a lot today. One of our sort of key strengths is integration of process equipment. The actual process suppliers are not interested in isolator technology. They're not interested in ergonomics. They want to give that to somebody else. So we will take that on as a company and make sure the systems are integrated correctly, safely, and they're very much usable by the process. So it's a big part of our application. We then move into dispensing isolators. Again, these are very, very common within every pharmaceutical facility. So we need to take that bulk material and dispense that down into whatever quantity to allow that to go to the next phase of the process. These can come in different designs. So the sort of standard approach on the top left there is a single chamber isolator, typically 4 gloves. We have an airlock which allows us to bring in products safely because that's interlocked from the process, and then we can carry out the dispensing operations utilizing weighing equipment, split butterfly valves, RTPs. So various types of systems are integrated into these designs to allow us to safely weigh and transfer materials out. We could go down to the multi-chamber route. So again, we may need separate chambers to bring in more materials. A lot of customers now ask us for storage chambers where we can bring in maybe 5 or 6 bags of product, so they can do a complete campaign without breaking containment. So again, we utilize similar designs for glove ports, filter housings, but the actual final product may look very different from the one we've done previously. Again, this is the bespoke application from Howard. So compounding systems, again, utilizing integration techniques. So we're using the same designs of isolators for dispensing, but we're now putting products into vessels. That vessel on the left-hand side is a mixing vessel with a WFI and maybe another material in there. So we're charging the raw material into that vessel. There'll then be a mixing process, and then that process will be transferred to, in this case, it was a filling line, and then filled into vials as part of the next part of the process. The top right shows an integrated mixer. So again, there's that word integration. So we can bring different solutions in for the compounding process. And this was a Polytron mixer, quite a high shear mixer that we've integrated into the isolator. So we can carry out some compounding steps within the system. An interesting one is tablet presses. So you're very familiar with tablet presses. There's a lot of equipment going on inside a press. They're quite often a square footprint, so they need to be looked at from an ergonomic challenge. And this is an example of a 4-sided isolator, so we can access all the various parts of the tablet machine. Most of that's for cleaning, most of that's for changing out the parts. But again, as the integration piece, we've taken the tablet press, we've done a bit of engineering work, we put the drive and the machinery outside of the isolator, so we just focus on the actual press itself inside the containment enclosure. Thinking of GMP, thinking of accessibility and clean down. Reactor charging. So again, part of the bigger manufacturing facilities, there's always that charging process into filter dryers, into vessels, into various systems. So again, these can take various shapes and sizes. We have the sort of multi-chamber version on the left with platform, because we've got to get that onto a reactor, which obviously means that the isolator itself is going to be tall. Or we have small scale, what we call bullnose isolators. So they have that pointy front, and this allows us to get close to the reactor, and we're handling small volumes of products. We're talking grams here, so we can charge that directly into the process. Another common process now is IBC charging. So for your tableting phases, for your blending phases, we need to get those materials into that IBC. We're not just handling APIs here, we can be handling the excipients, so the lactose or the different materials will perhaps need to be charged in stages. So we create like a sandwich within that IBC. So this particular example shows vacuum transfer systems from the isolator into the IBC. What you can't see on that image is there's a downflow booth where we're charging the excipients exactly the same way. You can just about see on the top left 2 black hoses. One of those hoses goes to the isolator, one of those hoses goes to the booth. So we can transfer the materials basically with a recipe into that IBC. That IBC then goes away to be blended and then carries on as part of the tableting process. Again, very bespoke, designed to give us the application the customer was looking for. We then move further down the line to the filtration stage. So again, integrating containment solutions with the pressure filter. The traditional way is to put the isolator on the outlet of the filter dryer, as you can see on the left. So as the blade of the filter dryer rotates, it then pushes the powder through the hatch, and then that goes into a container or a bag in the bottom chamber. The right-hand view is actually putting the filter dryer inside the isolator. This was a smaller volume filter dryer. It was a lab version, but that allows us then to access the material for sampling, for pack off, and to recover the heel from the plate, which drops into the isolator, and the operator would recover that through the glove ports. So as you can see, two different designs, but both doing the same feature. And then when we get out of the dryers, we then need to get into a contained pack-off system. I spoke earlier about the open continuous liner system. We've now put that into the isolator. So we've got a complete contained system, same operation, but through glove ports. Obviously, we can get a higher level of containment through the pack-off process. The application on the right-hand side was a 3-chamber version, so we had some cleaning capabilities in the right-hand chamber before the actual drum came out into the environment. And then we look at different ergonomic challenges. So in this particular example, we're using half suit technology. It's not a common technology. We do supply maybe 1 or 2 isolators a year with half suits. There are pros and cons of these systems versus glove ports. But for this particular example, we were integrating a cone mill. So this allowed us to take the product and ergonomically charge that into that mill. As you can see, it's quite a high system. So the operator with the half suit had a lot more flexibility to charge that process. We can then include the sieves, as you can see in the center there. So the L-shaped design allows us to be at a better height to charge the sieve. And then we've got a milling system on the right. That's a through-it hammer mill, and that's integrated through the isolator. And that basically milled straight through into the next part of the process. So again, that word integration, we're using it again, and it's bringing in all those different pieces of equipment. Chemical isolators. This is a special application for handling some really serious compounds. So as you can see, the design of the unit at the top left is very, very specific. I can't go into detail on this one, but it was very specific for the application. So it needed to be a big footprint to house some of the equipment that we're looking to test. And with this particular example, because of the chemicals that were being handled or potentially being handled, we had HEPA filtration as well as carbon filtration, including some backup systems, so backup fans, just because the customer needed that high level of protection should a fan fail or should a filter get blocked. We've got basically a safety system within a safety system here. So again, very bespoke and designed specifically for the application. Back to half suit technology. So this is a BSL-4 lab. Again, can't really go into much detail here, but this is a very specific design to handle BSL-4 compounds, and this particular unit utilizes a lot of aerosol equipment for their processing. You'll notice with this design we've used PVC rather than stainless steel. And again, this was a development with the customer over the number of years. It gives you vision. You can see through the whole process. So when we have operators inside the half suits, they don't feel enclosed, and it's more of an open feel when working inside the isolator. There's that word again, integration. So I just wanted to put some different examples on here. We've got different process vessels, the mixer that we talked about previously, tray dryers. So there's a number of systems that we can design into the containment enclosure. And then a new approach we're looking at is bringing in interconnecting systems. There is a unique design on this, which is very new to the business, and it'll be very new to the industry. So I'm sworn by secrecy on this, but we're looking at integrating a lot of processes into a containment line. And again, that gives a lot of flexibility when we want to transfer from process to process. We've got to get products into the isolators. So again, materials handling is very, very important. So we have a number of solutions from manual systems to automated systems where we can take the product drum. And as I said at the start, those drums come from various parts of the world from different suppliers. They won't change those drums. Them drums are fixed. That's what you get. So we have to find solutions to integrate those drums into the containment enclosure. And then finally, the sort of focus at the moment of the business is doing more turnkey projects. So this is part of a major project we're working on for the US at the moment. So this is 6 kilo lab facilities of this design. So the 6 of these that we're building. And again, we're integrating various different types of systems from process isolators with the tray dryers and lyophilizers. We have a rotovap system in there for some of the chemistry. We need to wash the bowls of the rotovap, so we've got a wash isolator. We've got the walk-in hood or the kilo lab, which again will house process vessels. Excuse me, turn that off. So again, that houses the reactor vessels and allows us to transfer basically between the whole system. And the prep isolator in this case is a dispensary which feeds into that reactor. So this has been a huge project for Howarth. The first one, as you can see here, has just been installed. So that as soon as the COVID restrictions were lifted, that was shipped over to the US. The guys have followed that, and we've just installed number 1, and number 2 will be heading over very shortly. The remainders are being assembled at Howarth as we speak. So this has been a real big project for us. Moving on to the turnkey facility, this is a raw material sampling facility. Again, very common within the pharmaceutical world. So as you can see, this is in a warehouse. You can see the bottom left. You can see the warehouse behind the facility. So we're bringing in a number of drums at any particular time to take representative samples before them drums go off into the production area. This particular system, we did the full clean room, we did the HVAC system, we have an isolator in there, we have a downflow booth, all the materials handling, roller conveyors, we did the full package. And this was a combination of the 2 businesses, the pharma business and the medical business, coming together and developing this system. So there is a little video which I'll just share, which you can see some of the operations taking place. So we took full responsibility for all the third-party materials. So the roller conveyors, I think we utilised a local company close to this facility. All the steel work, you'll see the duct work on top of the facility there. This is the downflow booth where we're taking the samples. So we have interconnecting doors between the facilities, so we can keep pressure areas, keep clean areas. So there will be a pressure cascade going through this facility to make sure we're maintaining containment throughout. So once we've taken the sample, the drums will either go out of the facility into an automated wrapping system, or we can keep them drums within the particular facility, and then we can take further samples utilizing the containment isolator. So a very flexible approach for material sampling. So then we just move on quickly to the EnviroSafe range. So this is an aseptic range that we've developed over the last few years, and the main product is sterility testing isolator. Our business is predominantly APIs, it's predominantly solid handling. So we've moved into the aseptic arena, as I said, quite recently, compared to a number of the suppliers that are out there that have been doing this for a number of years. But we have the sterility test isolator now developed, and this is really our main product for the aseptic environment. The cycle development has traditionally used the biological indicators, but we also have the capability of utilizing enzyme indicators. If you're not familiar with enzyme indicators, this is something maybe would be of interest, because this will give you immediate results. So the machine that's utilized to take the test takes about 10 seconds to give you the result, whereas BIs can take maybe a week and maybe longer. So that's a technology that might be of interest to some people out there. So if it is, then drop us a note at Inos, and we can give you some more information on that, because we believe that is a game changer for the gas cycle development of any aseptic system. So a couple little videos. So this is the gassing phase. So we use an ultrasonic nozzle, which gives like a bow tie effect, and that makes sure the VHP, the vaporized hydrogen peroxide, is transferred into the chamber, and we get full coverage. So we then go into the dwell phase. So this is where the VHP will sit within the chamber for approximately 1 hour. So we get full coverage and 10 to the -6 log kill. And then we have the aeration phase, which is when we're then removing the VHP from the process. And now we have a sterile environment, which we can then start the various testing activities. So you can see the VHP is removed from the chamber. That can either be taken out into the HVAC system, or we have a carbon filtration system which we can recirculate back to the room safely. We also have room systems. These are our mobile units, so we can decontaminate clean rooms, we can decontaminate labs. We use these a lot in hospitals, so we can wheel these systems in. And again, it's the same principle. We gas the room, go through the dwell phase, and then we aerate with the mobile towers that you can see in the centre. This system utilises a remote Wi-Fi tablet, so again, the operator can stay out. The operation can take place safely. We don't need to go into the room to do the process for obvious reasons. And then finally, we have unidirectional airflow systems. So again, these are very much a bespoke application. We can put the systems over existing filling lines. So we've got grade A supply, grade A air supply over the filling area, and that's utilizing HEPA filtration. The system on the left looks very straightforward, but again, we've got a UDAF at the top, which is providing grade A air over. In this case, it's bags of sterilized stoppers. So these stoppers will go into the vial filling process. This was actually part of a COVID manufacturing facility in the UK, and it was a fast track project because of the commitment the customer had made to the government to produce, I think it's 10 billion vials. This had to be in 3 months earlier than normal. So we worked very closely with the customer here to get the system up and running so they could do their testing on time, which I'm pleased to say we achieved. So that was a really important project to be involved with. And as we move on, we have to prove we can do what we say. So when we get into the high containment applications, we'll always do a containment performance test. So this is really a couple of examples of the results. And this is really important for me because this shows that we can give you the performance you need. And without that data, it's very difficult to say if the system you're using is going to be the correct system. So you really need to get the proof that these systems will work. So this was designed to 50 nanogram. And as you can see on the right-hand side, we achieved well within the target for that particular application. So this isolator was giving you the performance that it was designed to do. It's always very important to engage with the operators and engage with the SOPs, but at the end of the day, the system needs to be correct. And again, different types of systems. This was 10 nanogram, and if you look at the results, we were getting below 2 nanogram in a lot of cases. These are carried out by a third-party company. We don't do the testing; we support the testing, but this is done by a third-party company. Somebody like SafeBridge, you may have heard of, who would do the testing, and then they would provide the independent results. And even for the reactor charging element, where we're handling larger quantities, bigger volumes, we're still within the containment design that we were building to. So just to finish, I'll take you through some references just to give you a feel of who we work with. These are over the last couple of years, so you can see predominantly UK, US, Sweden, European projects. You'll see some names, familiar names there, your AstraZeneca's, SAFC. We've got GSK as one of our biggest customers. So we do work across the range of all the key pharmaceutical companies, various different types of applications. MSD Singapore is a new project where we're looking at quite a big facility over there. We've actually been using the team from Jacobs in India on this particular project. So yeah, it's becoming a real global operation. And then finally, a couple of testimonials. We're always very proud on how we deliver our products and the support and the feedback we get from the customer. and this was the one I referred to earlier about the COVID project, where we had to find a better way of delivering the system quicker. We managed to do that. So yeah, that takes you through the business. I feel like that's gone quite quickly there. So hopefully you captured a lot of that. And if there's any questions, then I'll be happy to answer.
Thank you so much, Paul, for the presentation. The applications, especially, were very interesting, and I'm sure everyone who has attended will benefit from this. So, Paul, we have some questions from the audience today. So, can you please? The first question is, can you please elaborate the difference between isolator, ORABs, and CRABs?
Yeah, it's that's becoming very interesting because traditionally your CRABs are your closed RABs, and that's more the UDAF technology, where we're providing the grade A environments over the process. The ORABs, the open RABs, again, that grade A airflow would go back into the room and get recirculated. But what we're seeing now is that closed RABs are starting to morph into isolators. And again, that's just because of the hazardous materials. So they become more of a single pass system. We have to take the air away, and that either gets conditioned or that gets removed from the room. So it's a very interesting comment, because depending on the customer and the application, when you use the word isolator, they sort of don't like that, because they're not after an isolator, they're after a RABs. A RABs is a restricted access barrier, which is exactly what an isolator is. So it is becoming pretty much the same technology as we see the products becoming more and more hazardous. So again, I'll throw it back. It's always part of the process and the operation. If you can accept more of an open system, or it becomes a closed system, and then it's a RABs versus an isolator. So yeah, it's becoming more of an interesting discussion now. Probably didn't answer your question, because I'm basically saying there might not be a lot of difference nowadays, but yeah, it's just one that I think we'll keep progressing.
Okay. Thank you so much, Paul, for that answer, and I hope that answers the question. So coming to the second question, what are the design parameter consideration for OEL 6?
Well, I think I focused on 6. So it's OEB 6, not OEL 6. So I focused on that earlier on in the presentation. So this is where the high containment isolators are an absolute must. So it's using glove box technology. It's thinking about the integration of systems. As I started off on the presentation, materials in, materials out is absolutely important. So the isolator itself will be designed to OEB 6, but we need to make sure that split valves or RTPs or transfer systems have the same performance, because that could be the weak point. And it's really important to get the OEB 6 and the OEB 5 correct, because we're talking, what did I say? I think it was 60 million parts of a grain of sugar. We've got to make sure that's contained. So yeah, any application which is in that band for me will always be a containment isolator.
Thank you, Paul.
And just to finish, and that's where the bespoke element comes in, because we don't have an off-the-shelf solution. We have to design each application for against all the different processes that they're looking to do in that system.
Right. So basically, if you can just share the process through mail with us, and maybe we can work out and get back to you in detail for that. Yeah. So coming to the next question, this is from Mr. Durga Ganesh. If a negative pressure is maintained in isolator, how to handle sterile powders in isolator, and recommendation for the same?
Well, when you move into the sort of sterile applications, that does become traditionally a positive system. But we're seeing that not very often, but there are the cases where that powder is hazardous. So we have to look at maybe a combination of positive pressure, negative pressure, or in my opinion, it needs to be a negative pressure system, because the operator obviously is of key importance. So we have to look at different designs. It's a very much a combined approach, but sterile powders should be handled within a positive environment. But if there's a hazard there, then that's something we'd have to take a real view on. Again, it becomes a very much a bespoke application, depending on that particular powder.
Okay. Durga sir, I hope that answers your question. Coming to the next question, can humidity be controlled inside the isolator, like 20% RH to 30% RH?
Yeah, absolutely. So again, that's quite a common request now. So the input air or nitrogen is often used. So we look at dry compressed air or nitrogen. Nitrogen will give us quite low humidity within the system. So it all depends on where you are in India, because the humidity could be different at different parts of the country. So these are the kind of things that we'd have to look at. But once we get the right conditions, then the isolator would monitor those conditions. So we'd use instrumentation like oxygen analyzers. So for instance, we know we're at a certain percentage. We'd use humidity system so we can measure the humidity, and if it does go out of control, the system would alarm, and that allows the operators to make any changes. For any really tight bands of humidity, we can put on dehum systems, so the actual air would be treated before it gets into the isolator. So this would be an extra piece of process equipment where the air would feed through. It's like a desiccant wheel, and then humidity is controlled within that system before it goes into the isolator. So yeah, there's various ways of doing it. Nitrogen's always the preference, because that we know that's a dry gas, but it depends. Some sites don't have nitrogen, or they have to bring in nitrogen tanks. But dry compressed air can also give us the level of humidity that we need inside that chamber.
Thank you, Paul, for taking that question. Nilesh sir, I hope that answers your question. Coming to the next question, that is, if Paul, can you just brief regarding VHP validation for isolators?
Yeah, so I touched on this earlier. So the gas cycle development is obviously a real key part of the process. So we would do the cycle development test utilizing enzyme indicators. So we do that in our factory just so we can prove the system will give us the 10 to the -6 log kill that we need. The industry generally utilizes BIs or biological indicators, because that's a validated process. So when we take the isolators to the customer site, we would then do the test using BIs. That does take time, as I said previously. It can be, I don't know, several days for those indicators to be tested, which can slow down the process and the operation. So we are recommending the enzyme indicators more and more now to speed up that process. But as I said, we generally focus on sterility test isolators at the moment. That's our main product for the aseptic range. So everything that we do has been pre-validated. We have all the test data. We have all the results now for us to give you the right system.
Okay, thank you, Paul. We have two last questions. Next one is, if there is any other special requirement to handle steroidal products.
I think this ties in with the sort of high containment applications. So that's where a steroidal product would come in, because it needs that that level of protection an oncology drug would. So yeah, it would be designed around an isolator system, and again, depending on what the process is, but that would be the recommendation. We wouldn't go outside of an isolator for those type of products.
Okay. Next question is, what should be the personal protection level for handling steroidal products in biohazard and LAF cabin?
Well, again, I wouldn't personally recommend that they're within a laminar flow system, because that indicates more of an open environment. And we need to keep that product away from the operator. So in my experience, those type of applications are always used inside an isolator. I've not personally come across a steroidal product within an open environment.
Okay, I hope that answers the question. So the last question for today, for placement of BIBO filters, what design considerations should be taken in?
Well, the bag in, bag out technology is generally utilized within the downflow booths. So we have the bigger filters within a safe change housing. So that's where we would use the BIBO system. Within our isolators, we move to push filters. So there are smaller filters, but we call push because we would push the secondary filter into the primary position, and the primary filter then gets removed to waste. By doing this, we're keeping the containment within the isolator boundary, and it's a real safe way of changing the filters out. Some customers ask for filter housings as the bag in, bag out method to be remote to the process, which is achievable, but you have to be mindful that we're going to have a contaminated piece of ductwork from the process system to the filter system. So it's always a preference to keep that filtration as close to the process as possible, because if then you transfer that powder through a duct, you've got a piece of duct to clean. That duct could be 10, 20 meters, 100 mil diameter, before it gets to the filter housing, which is a bag in, bag out system. So that's something just to be aware of, that we don't want to transfer that product further down the line into a filter system. So if we can keep everything as close as possible to the process, then that's the best way of designing the system.
Thank you so much, Paul. I think we have answered all the questions that we had received. I would like to take this opportunity to thank everyone for your time and attention, and taking time out to attend this wonderful webinar on containment solution. I'd also like to thank Paul for your time and this presentation, which was very informative, and hopefully the applications and the case studies that you have shared with us will be very useful for everyone. And if there are any other further questions, you can always share it on mail to info@inos.in and seema@inos.in, and we'll come back to you with a suitable solution. I would like to thank everyone again to attend this webinar. Thank you.
Yeah, thanks, Seema. Thanks, everybody. And hope you all stay safe.
Thank you, Paul. Yeah.
Thank you. Bye-bye.
Bye-bye.